I am particularly pleased and honored to have here with us today David Quammen, American science writer and author, of course, of Spillover, which today is a sort of worldwide bestseller. And in 2012, he was already raising awareness on topics concerning a possible pandemic virus, which, of course, are very relevant today. But David Quammen is author of several books. Some of these are about evolution and biogeography, for example. And the last one he wrote is The Tangle Tree, published in Italy by Adelphi, l'albero intricato. And he will give a special lecture based on this book at the Genova Science Festival next October 24th. So first of all, thank you very much, David Quammen, for being with us today.
You're very welcome, Francesco. Very good to be with you. Nice to talk with you.
So the tree, the title of your book refers to, of course, is the evolutionary tree. But what is it about this tree that is tangled?
Yes, the evolutionary tree of life. And tree of life is sort of an old image, goes back to the Bible, but it was Darwin who really drew the first evolutionary tree of life, pointing out that the shape of the history of evolution on Earth resembles a tree, with major limbs branching from a common trunk, branches diverging from the limbs, diverging, always diverging. And the classic Darwinian tree throughout most of the 20th century reproduced that idea, diverging, limbs always diverging, so that inheritance would occur, evolutionary change would occur over time as different lineages of creatures, they left their offspring, and they changed, and sometimes they were separated and different populations diverged into different species. What changed radically in the late 20th century, thanks particularly to the discoveries and the legacy of Carl Woese, my central character, was the recognition that sometimes limbs and branches converge, they come together. How is that possible? It was supposed to be impossible, but it's a phenomenon known as horizontal gene transfer, the actual movement of genes and sections of genomic information, sections of DNA and the other molecule of heredity, RNA, could actually move sideways from one species into another, from one genus, even from one kingdom to another, great limbs converging on this tree. And people say, well, how is that possible for genes to move sideways? The most simple way I could explain that is one of the terms is infective heredity. Viruses, for instance, can pick up stretches of DNA from one creature that they have infected and then reproduce that DNA as the viruses reproduce and then carry it to another creature if they infect that creature and drop it. So that's one of the mechanisms of horizontal gene transfer. Viruses picking up DNA in one kind of creature and moving it to another horizontally. So this horizontal gene transfer represents the entanglement of the tree of life, which is why my book is called The Tangled Tree.
And in this book, you tell the stories of many great scientists that contributed to build this new conception of the tree of life. And Carl Woese is a sort of a neglected hero, maybe, of this revolutionary conception of the tree. It is not well known, maybe, in the wide public. But he discovered in 1977 a whole new group of living beings, the Archaea. So what are the Archaea and why do they matter?
Yes, and you're right. Woese, W-O-E-S-E, this fellow, this microbiologist, molecular biologist, a little man in Urbana, Illinois, in the late 1970s, began this revolution. And I refer to him sometimes as the most important biologist of the 20th century that you've never heard of, because most people, even many biologists, haven't heard of him. Well, so in 1977, he and his group, including a postdoc named George Fox, made this discovery using methods that they had invented. And I can talk about the methods, but it involved essentially looking at certain very basic molecules that are in all forms of cellular life and sequencing those molecules, that molecule, and then comparing genomic sequences. So you could compare a bacteria with a horse by looking at this particular molecule. And so they started doing all these comparisons, and they looked at a lot of bacteria and comparing these molecules, and they discovered that some of these things that science had been calling bacteria for 200 years, 300 years, were not bacteria at all. In fact, were so radically different in this particular molecule that they're more closely related to us. These little one-celled things that look like bacteria through a microscope, they're more closely related to us in terms of the way their cells are built than they are to bacteria. And it was thought at the beginning that they might be the oldest form of life, a new discovery of the oldest form of life, and therefore they took the name Archaea. And that was Wose's first great discovery.
So,
and of course, people said you're crazy at the beginning. People all over the world said, "Oh, that's garbage, that's baloney." But then gradually they looked at the evidence, and other people confirmed his discovery, and it became recognized as true.
And so thanks to Carl Wilson, now we know that at the basis of the tree of life, there are not two groups, as we thought before, that were eukaryotes and prokaryotes, but there are actually three groups. There are bacteria, archaea, and eukaryotes. And prokaryotes are not even there because it is a group that doesn't make much sense nowadays.
Right. Right. That was the old category, prokaryote. And prokaryote contained everything that was a unicellular creature with simple cells without complex internal cell machinery like a cell nucleus or mitochondria, these internal organs that package energy. So that's why people had looked at these things and said, well, they're all bacteria. So there was the bacteria, the prokaryotes, and the eukarya, complex creatures with complex cells, animals, plants, fungi. And those were the two big limbs of the tree of life. And then Wose's discovery of the archaea put a third great limb in the middle. And it proved that the category of prokaryote was useless because these two great limbs both could be defined as prokaryote, and yet they were very, very different in all other ways. So from two limbs, it went to three limbs, and the story becomes even more complicated from there.
It is even much more complicated because these methods you were talking about, so this molecular phylogenetics, today is applied in a lot of labs. And recently, some researchers suggested that the roots of this tree of life might still be two, not of course because they don't believe in the existence of archaea. Of course, archaea are there, but the point is that archaea and eukaryotes might have had a common ancestor. And this was also an idea that Carl was discussing with a famous scientific rival of his, which was James Lake. And is this debate between the two or three roots of the tree of life still there today? Is it still an open debate today?
The debate is still open, yes. And you mentioned James Lake. He led one school of thinking about this. He actually thought that there should be four major limbs drawn on the tree of life. Now people are saying not four, not three, there should be only two, and our branch of our limb of the tree of life doesn't exist. It's not a major limb. We eukaryotes, all animals, plants, fungi, we are a minor branching from one of the two major limbs of the tree of life, bacteria, and the archaea. So as the tree is drawn by some scientists now, there's only those two, bacteria and archaea are our ultimate ancestors, and all of the eukaryotes are simply a secondary limb branching off of the great limb of the archaea. But that's controversial also, wonderfully controversial, with brilliant people on both sides.
This is definitely a very hot and interesting debate. But you mentioned horizontal gene transfer as the main mechanism that makes the tree of life tangled. But I would like to ask you, is it the whole tree of life that is tangled in the same way, or are different parts of the tree of life tangled in different ways? I mean, this is something specifically concerning bacterial and microbial life, or does it applies also to the other parts of the tree of life?
Oh, this applies also to eukaryotes. It applies not just to bacteria and archaea. Horizontal gene transfer is very common between different kinds of bacteria. We could talk about antibiotic resistance, which comes in there, and archaea. It's very easy and very common for them to pass genes sideways between completely different strains or species of bacteria. And some scientists argue that yes, horizontal gene transfer is very common in those single-celled creatures, but it's almost insignificant in complex creatures with genomes that are protected inside cell nuclei. But then more and more work in the late 20th century, following the legacy of Carl Woese, work by people such as Cedric Fichot, a very interesting scientist who works on detecting horizontal gene transfer, and a number of others that I mentioned in my book, have shown that there are genetic elements that have been moving sideways also into complex creatures for as long as we have been complex creatures by different mechanisms, but also including what I mentioned, viral infection. For instance, 8% of the human genome consists of viral DNA that has been brought into us, into our lineage of animals and then mammals and then primates and humans over the millions of years, accumulating to the level of 8% brought in by retroviruses. Viral DNA brought in by retroviruses, retro meaning they work backwards. They have an RNA genome, they create DNA, and they patch that into the genome of the cell they have infected. The HIV virus moves backwards and patches its genome into the cells of our immune system. But these endogenous retroviruses move backwards and patch the DNA into the cells of our reproductive systems, into eggs, human eggs and sperm, and the stem cells that make eggs and sperm. And when that happens, it becomes heritable. And so we have this inheritance, 8% of the human genome, including some genes that are absolutely essential for human life. They have been adopted for use in mammals. For instance, one gene that creates a membrane between the placenta and the fetus, utterly necessary for human pregnancy. That gene is a viral gene that used to create an envelope around a retrovirus, and now it creates an envelope around the human fetus.
Well, this is very fascinating. I think we can say that there are also good viruses, right, that help us doing our fundamental functions, biological functions. I mean,
absolutely, and that gene that I just mentioned, it comes from a virus that has been very beneficial, that particular retrovirus. There are other retroviruses, endogenous retroviruses in the human genome. As a matter of fact, I'm just finishing this week writing an essay for National Geographic magazine on this subject, on the ultimate evolutionary origins of viruses. Where do they fit on the tree of life, if they do fit at all? And in what ways have viruses been important in positive ways in the history of evolution and been beneficial even to us humans? So I'm just editing that piece this week. There are a number of ways in addition to what I just mentioned.
I will definitely have a look at that essay. So in 2012, you wrote Spillover, and you reported about scientific voices warning about a possible pandemic. Now in the Tangled Tree, you write about horizontal gene transfer, which is the mechanism through which antibiotic resistance occurs. You just mentioned it. So I would like to ask you, is it, first of all, what is exactly antibiotic resistance, and should we consider it the next big health issue alongside this pandemic and climate change for the years to come?
Well, antibiotic resistance is certainly a major health issue. I don't know that it will ever be as as devastating in a short period of time as COVID-19, but it's something that is attracting attention of medical people, and it kills antibiotic resistant bacteria, bacteria that have evolved or acquired resistance to particular antibiotics, tetracycline, streptomycin, penicillin, whatever. Those kinds of bacteria kill tens of thousands of people a year. I think tens of thousands of people just in the US, and probably it adds up to hundreds of thousands of people around the world, but I would have to check the exact numbers on that. But it's a very serious problem. How does it occur? It occurs in two ways, and one of which has been long understood, and the other of which has been neglected. And I describe the neglected one in my book. The way that's been long understood is that if you have a strain of bacteria, for instance, Staphylococcus aureus, a very dangerous kind of bacteria, and those infections are treated with antibiotic over long periods of time, and the bacterium is exposed to this antibiotic, then it can gradually acquire resistance to that antibiotic, just the way a population of grasshoppers can gradually acquire resistance to a pesticide. If you have grasshoppers in your field and you spray the field with DDT, then you'll kill 99% of the grasshoppers, and 1% left will happen to have some natural resistance to DDT. And the following year, you'll have a field full of grasshoppers again, but now they will be resistant because they will all be descended from that 1% that was resistant. Same thing happens with antibiotics, but it takes time. That's a slow, incremental kind of evolution that involves simple mutations in the genetic code that add up gradually over time. If a bacterium is hit with antibiotic over and over again, these mutations add up, and finally you have a resistant strain of bacteria. What wasn't recognized, and I talk about it in my book, until some discoveries, first by a Japanese scientist named Watanabe, is that bacteria can pass the genes for resistance sideways in an instant by horizontal gene transfer. So it might take years for Staphylococcus aureus to evolve the old-fashioned way, resistance to streptomycin. But then that resistance, the genes for that resistance, could pass from a population of Staphylococcus aureus, possibly in an infected wound of some person. If it was infected also with streptococcus, not staphylococcus, streptococcus of some sort. And the gene could pass in an instant from Staphylococcus aureus into this completely different species of bacteria. And that bacteria then, those bacteria then would be resistant. So that's how antibiotic resistance has swept around the world so quickly. And that sort of passage apparently particularly happens in hospitals, where you have a lot of sick people, you'll have a lot of different strains of bacteria, you have a lot of antibiotics that are being used. So it's a situation in which horizontal gene transfer can occur.
The very last question. What do you think our world community should do in order to get prepared to antibiotic resistance, and maybe not to repeat the same mistakes maybe we did with this pandemic virus, because maybe we were not that prepared after all, despite all the warning people like you also.
Yes, yeah, and these are two very different problems. But the one thing that they have in common is, to prepare better for them, we need support for science. We need to end with the science denying, which is, which is almost a pandemic of its own right now, particularly in my country, to some extent in your country, to some extent in the United Kingdom. We need to get past that. We need to trust scientists. We need to trust public health officials. We need to support science. We need to support it financially as well as intellectually. We need to get it into the schools. We need to be teaching our young people science, evolutionary biology, and other forms of science. We need to be doing that, so we're raising new generations of brilliant scientists upon whom we can depend and whom we can trust. that's the beginning of it. And that's one thing that these two different problems have in common.
David Quammen, thank you very much for being with us. It's been an incredible pleasure chatting with you. And
a pleasure chatting with you too, Francesco.
And enjoy your Genova Science Festival.
I look forward to it very much on the 24th. Yes, being part of the Genova Science Festival. It'll be an honor and a pleasure.
Okay, thank you very much, and thanks for your work once again.